Well, thank you very much for the opportunity to present today. Probably what I'm going to present is a little bit different from many of the other presentations you've seen, in that we are a combined academic and governmental institution, and we focus very much on bridging the gap between very early phase developmental work and getting these into an early clinical trial. So, which button is forward? Okay, that's not the right. Green arrow. So am I just being incredibly stupid here? Oh yeah, the big green arrow, thank you. Okay, the work I'm going to talk about today is based in Edinburgh. The pictures at the top are probably the ones you're maybe more familiar with Edinburgh, but we are concentrated in two centres in Edinburgh. The Scottish Centre for Regenerative Medicine, which is an innovative building run by the University of Edinburgh and the NHS, where we have brought together the ability to develop new therapies, then to translate them, and then actually to manufacture them for early phase trials. The second science centre on the right is the new SMBTS centre, which we're just about to occupy, which is a large blood transfusion centre, but also has ATMP manufacturing. So we work across two different sites in Edinburgh. So why on earth are we talking to you today? Why are we doing cell therapy in the blood and tissue service? Simplest way to think about this is every single bag of blood that is transfused is a cell therapy. It's been collected from a donor, it's been processed, it's been safety checked, it's been formulated, it's been stability checked, and it's been delivered to the patient that needs it. We took this about 20 years ago, and this was really pioneered by Professor Mark Turner, who leads us today, in that we did our first, what was then not called an ATMP, but it is an ATMP, in that we were making dendritic cells to treat breast cancer patients in early phase trials nearly 20 years ago now. And this is an expertise that we have built on and we have refined over the years to now provide the unit that we are, where we specialise in these translation services. We are a little bit more sophisticated these days. We are no longer doing stuff in plastic flasks with pink medium. We have really a very well-equipped system now where we use the latest cell processing equipment. We work to full GMP. And just as an example that there's really nothing new in this earth, we're currently treating patients with cirrhosis with their own macrophages made out of monocytes. And this process is really not that different from the dendritic cells of 20 years ago. So what is cell therapy? Well, there's many ways to look at it, but you can slice it this way, which is it's the facilities to actually receive the tissue, qualify it, process it. It's the biological expertise to understand the science behind it and how these cells will grow. You can see here there's three different examples of the cells that we grow in the middle column. On the top, these are corneal epithelial cells. These are taken from deceased donor eyes, and we grow the stem cells from these on a membrane for transplant into the eye. In the middle are pancreatic islets. We are one of the pancreatic islet transplant centres in the UK, and actually a lot of what we've learned about how to process tissues into complex mixtures and into cell therapies has come from this. And at the bottom, there's an example of a much more complex long-term product. These are T cells that are used to treat tumours that result from immune suppression in patients. The other thing you have to have, and this is the thing that we use, this infrastructure we've developed really over 70 years, is the quality management structure, which is we have got the capability to deal with the licensing, with the regulatory compliance, and also most importantly in the middle picture, we have the ninjas who run the clean room. They're really important too. So how are we set up? We're really set up for three different areas of translation. One is basic biology. It's run by Dr. Jo Mountford, and she really is a stem cell biologist that leads a really strong stem cell team, both induced pluripotent cells, ES cells, and hematopoietic cells. In the middle, we have the cellular identity function. This for us is a really important area that we work extensively in translating what are often very complex research assays into things that can be used to release cell products for use in patients. Then we have a SCRM, or GMP translation and manufacturing space, where we actually make the products. I think it's very important to say these are all aimed at very early phase trials, phase 1, phase 2, and this is our aim, which is to get things into clinical trial. We are not looking to then develop it into the large-scale trials. So this is where we are based. We are based at the SCRM. The facility here is the beige part right in the core, and this is a 7 clean room facility that we share with Roslin Cells, and we have 4 clean rooms and they have 3. But most importantly, our university and medical service translation labs are right adjacent to the facility, so we can literally make it across the corridor and then get it into the clean room and make those therapies. This is the site that we work on in Edinburgh. The massive building you see in the middle there is the Royal Infirmary of Edinburgh. This is an excellent site for doing early phase trials. You can see here we have a variety of early phase clinical trial units. So we have the CRF, which is a specific hospital set-up ward for doing early phase trials. We have the Experimental Imaging Centre here. Much of what we do is around tissue regeneration. And we also have the ability to manufacture on the same site and then give the cells to those patients on the same site. That may sound trivial, but when you have to validate carrying something across the car park rather than having to then put it a long way to get to your first patients, it's really rather important. As I said, we're really a partnership organisation. What we aim to do is to develop some of our own therapies. Much of it is done in collaboration with our academic partners. And there's a number of different therapies that we have developed over the years. I've already touched on the islet process. We are one of the biggest islet transplant centres in the UK. And we are very much using the Edmonton process, although we aim to refine it, to treat patients with severe type 1 diabetes. In the middle, we have our T-cell product. This is an EBV-specific T-cell product. Again, very much in the tradition of what we do, this is an allobank. In fact, now we make these cells with donations from New Zealand, so we ship them from the other side of the world. We manufacture them into EBV-specific cells, and then these are used right across Europe in treating patients with PTLD. Carneal stem cells I've already touched on. This has just finished its phase 2 trial, which was our controlled trial. Phase 2, where we've looked to treat ocular surface disorder with epithelial stem cells. Then on the bottom, we have our more recent things that are just coming to development. We are banking again, allobanking MSCs from a variety of clinical sources. This trial here is unique. It's its first in man. This is a macrophage product where we take CD14 positive monocytes from patients with cirrhosis. We turn them into macrophages which can remodel the fibrosis of the liver, and this is now into its early phase trial. And this was preceded by a trial of CD133 cells, the realistic trial where we took hematopoietic stem cells also to look to try and treat cirrhosis. So what we've got here is a number of different capabilities, a number of different therapies that we have developed in conjunction with our partners. What's our philosophy about developing cell therapies? We try and start with the end in mind, and that is that we do not generally start with non-GMP compliant components. We normally try and go to a fully GMP product, a fully enclosed product, and we also try and build things in blocks, the sample reception, cell isolation, the cell manipulation, and then analysis. So these are building blocks that we can apply to every single product that comes through our doors, and we can then fit that to whatever the cell product is. How do we support this? As I said, we are a government institution and an academic institution. We are part of the NHS, the National Health System in the UK, and we have a remit to try and improve health. And very much what we're aiming to do here is to understand the feasibility, how you deliver new therapies, and to get these into the early phase trials so these can then go on to larger, more industrial organizations after that to try and treat many more patients. So we bring a lot of core facilities to this. We have staff, we have microbiological surveillance, we have manufacturing, we have cell analysis, and we also have a core development function. And when you put all that together, that can be applied to a number of different scenarios to try and accelerate things into the clinic as quickly as possible. So I just thought I'd finish in the last couple of minutes by showing a couple of the therapies that we have in development. Right now, we are looking at endothelial cells for blood vessel repair. This is an autologous product. This is something that is now just about to go into, I wish I didn't do that, into a clinical trial. We have here now developed a completely xeno-free product. So although this product is in its very first phase, this is completely xeno-free, it's highly reproducible, and that means that this thing can scale very quickly into larger phase trials as required. Similarly, we are developing mesenchymal cells. I think it's been really telling. A lot of people at this conference this week have been talking about not bone marrow cells, but ones from other sources, and that's something that we are very much interested in. Again, we are developing an allobank of mesenchymal cells that would be available for a number of different clinical trials from multiple tissue sources. But what we've done is we've taken our expertise to develop a single xeno-free process for use with mesenchymal cells, and that can be applied to every tissue that we have examined so far. And we aim to bank these from allo donors at scale, and then these can be used in a number of different clinical trials. Similarly, we are involved in something called the Novozyme project. You may or may not be familiar with this. This is a very large consortium which aims to make in vitro red blood cells from a pluripotent source. And this is something now that we're in the last stages of translation, the scale of the progenitor cells. This is already a very complex ATMP, and then we have at least 3 distinct cell stages that we have to qualify and we have to get right to produce these in vitro red cells. And finally, our EBV-specific CTLs for using in treating PTLD. This has been made in a very intensive, traditional way until now, similar to, there's a couple of other sites in the US use a similar technique. And that's using virus-infected cells. It's a long-term process. It takes a long time to grow the cells. We've now changed this into an antigen-driven, antigen-specific T-cell selective process, which then has shortened the time by 2 months to make each line. And most importantly, we're aiming to make the first allobank of these cells. So this is going to be a viral-free allobank, again, to treat patients as required. So I think I've fulfilled my remit of getting this done in 12 minutes. Thanks very much for your attention. I hope that's been of interest, and we'll be around for the rest of the conference. Thanks.